All right, so good afternoon, everyone. And it's my great pleasure and honor to welcome all of you to the 2020 Distinguished Visiting Scholar Award Ceremony. And thank you all very much for joining us on this very special day for our Department of Biostatistics at the University of Michigan, and for being here to celebrate the legacy and excellent contributions of our dear colleague, mentor, and friend, Professor Michael Boehnke, through the presentation of 2023 Michael Boehnke Distinguished Visiting Scholar Award. I would like to extend the gratitude to a few people who made this event possible: Mandy, Kyle, and Dave. And we are very fortunate to have this amazing staff team in our department. We really appreciate that. Appreciate their tremendous support and effort over the years, especially for this terrific event. Thank you all very much. And here's the agenda for today's event. Brahma and I will start with a brief introduction, followed by Professor Sylvia Richardson's talk on scalable Bayesian inference for high-dimensional genomics. And after that, we would like to invite everyone to the reception, which will be held in the assembly hall across the hallway. And hopefully our dear chair, Brahma, has approved the alcohol for those who are interested in. But nevertheless, I'm pretty sure we will serve wonderful food and refreshment there. And now, please allow me to spend a couple of minutes to briefly share with you the history of this wonderful award. As all of you know, Professor Michael Boehnke is a research pioneer and leader in the field of statistical genetics. His impact has been profound in both biostatistics and beyond in our profession. He is always joking with us in many occasions that he's probably one of those who has been in the department or been around for the longest. You know, that always makes me think maybe one day I could proudly claim similar things in this department. You know, this is my first job, so I came here right after I graduated. I've been working here for 15 years, very hard. That is very long, so probably one day, who knows, right? But yesterday, after I talked to Mike and confirmed about the date when he joined the department, I found that it will be never be possible for me to beat that record anymore. Mike joined the department in 1984, January the 1st, so he emphasized that. That is when I was just 3 years old. And he has dedicated almost 40 years to this department. Given that wonderful number, I think he definitely deserves a big round of applause. As his colleagues and academic families, we are so fortunate and grateful to have him in Michigan Biostatistics. He is an outstanding statistical scientist, a dedicated and admired advisor for numerous trainees, and a very generous mentor for many junior faculty members, including me. He is the director of our Center for Statistical Genetics and the director for Genome Science Training Program, and he has served as our graduate student admission committee chair for many years. Over the decade, his vision and leadership have shaped our research, training, education, and culture of this department in many fundamental ways. This award was established in 2022, named after Mike, to honor his extraordinary contributions to the Department of Biostatistics at the University of Michigan and to the broader field of biostatistics and human genetics. And this visiting scholar program aims to promote interchange of new research ideas and collaboration between the visiting scholar and the faculty and the trainees at UM Biostat. The visiting scholar will offer short courses, seminars, and meetings with our trainees and faculty. The award is given annually, and last year we are very honored to have the very first recipient of this award, Professor Ken Lange from UCLA. He spent 2 weeks with us visiting our department and shared many of interesting topics and ideas with us that include algorithm and its applications, how to play the art of scientific writing, and copular-like distribution for statistical modeling, etc. So this year, we also received many nominations for the award candidate. The biostatistics faculty voted together, and our 2023 award goes to Professor Sylvia Richardson from University of Cambridge. Congratulations, Sylvia. With that, I'm going to pass the floor to our wonderful department chair, Professor Brahma Mahaji, to introduce Professor Richardson, her research, her leadership, and her life.
Thank you so much, Lou. By introducing Mike, you also revealed your age. But I just want to take this moment to really reflect on the journey of two remarkable individuals. And I think I love biostatistics because we have these remarkable individuals and they are so nice and kind. They don't have a little bit of ego or who we are. They're so down to earth, and they are actually, I was afraid to talk to Mike and Rod and Sylvia for the longest time in conferences, but then I realized I missed out. They're wonderful human beings. So before I say anything about Sylvia's extraordinary career, I want to invite Mike and Sylvia both for the presentation of the Distinguished Lectureship Award. For those of you who are online, we put a copy of the photo of the plaque on the slide so that you can see it clearly. I'd like to invite Mike to say a few words.
Thank you, Brahma, and thank you all for being here. Thanks, Lou, for that wonderful introduction. I almost don't want to say anything after that. It was so nice of you. Sylvia, thank you so much for being here. I'm looking forward to a third wonderful talk. To Brahma, the department, and all the friends of the department who've established this award, thanks so much. It's really an honor. And Sylvia, it's truly an honor to have our names linked together in this way. So, welcome.
So now it's my turn. I have to say that Sylvia and I have become very good friends over the last 1 year because she was my host during my sabbatical. But I want to say honestly, I did not nominate Sylvia for this award. And I did not even participate in voting, just to show any kind of implicit or bias. But I have really enjoyed knowing Sylvia as a person, as a wonderful scholar with tremendous insight and intuition into a wide array of statistical problems. So we had a wonderful dinner with Mike and Sylvia and Betsy in our home. And then this photo was taken when Sylvia, just after Sylvia gave the presidential address for the Royal Statistical Society. So her smile is very wide after talks are over. And so you expect to see that in the reception as well. So I want to say that, there are very few statisticians that I admire so much. Because Sylvia has not only held many, many leadership roles in a department, in a community, in a professional society. As her daughter said, that this petite woman from France is a commander of the most excellent order of the British Empire medal. It's unbelievable how she conquered all the struggles and kept achieving milestones in a very natural and fluent way. She's the winner of the Guy Medal in Silver. She's an IMS medallion lecturer. She has received an honorary degree from University of Oslo She's fellow of AMS, IMS, ASA, ISBA, and Academy of Medical Sciences. So during COVID, she was co-chairing the Royal Statistical Society's task force on COVID-19. And so that made an incredible influence on how data were analyzed, and we got to hear a bit of that during her lectures here. But her presidential role as Royal Statistical Society was remarkable because she did not anticipate, none of us could forecast the COVID-19 pandemic, but she provided tremendous leadership to challenging issues in COVID-19. I want to show you that she's a very impactful scholar. Her citations, more than 40,000 citations, is incredible. But to have all those citations, mostly from statistical papers, not really collaborative papers, is really, really remarkable. Those of us who are in the business realize that how hard it is to get a statistical paper on Bayesian analysis of mixture models, the pioneering paper with Peter Green, in having nearly 3,000 citations. And also non-Bayesian work in terms of spatial processes, how to test correlation of two spatial processes, something simple but elegant, used in geography, used in ecology. So I became a fan of Sylvia. Actually, I graduated in 2001, and I became a fan of Sylvia by this paper, which does not have 3,000 citations, but 300 citations. Some of them are mine. And so it is on variable selection in case control studies. And there are very few papers on Bayesian analysis of case control studies, and this was one of them, and it was extremely clever, and I became a fan. But when I was visiting, I saw how loved and admired Sylvia is by her family. This is a picture with her kids. And then on the left-hand side with two Royal Statistical Society presidents, Deborah Ashby and Sir David Spiegelhalter. And I had the pleasure of seeing her during her retirement party at my home. This was my sabbatical home in Cambridge, I miss it. And how admired Sylvia is by her community. And wherever she has gone, she has created a community. Be it at Imperial College, be it at MRC Biostatistics Unit, she just did not push her own agenda, own research agenda. She brought people together. This is a tremendous skill set that I profoundly, profoundly admire in Sylvia. Building communities during Sylvia's visit. We have had the pleasure of having Sylvia for the last 2 weeks almost here. And I know that you must be tired, but I promise that the trip up north is going to be relaxing, and we are not going to ask you to give a talk in Traverse City. But she really bonded with faculty, she really bonded with students, and immediately develops a conversation and a research connection. So we are very, very thankful for your time, because 2 weeks is not easy. I also want to say that this French-British style of Sylvia influenced me profoundly. Her modeling in terms of statistical modeling, but also fashion style, was inspiring for me. So this was Sylvia just before giving her presidential address, and I just noticed her shoes, those red flats. And so I came back to US, and I got the same red flats, and both of us are wearing those two red flats today. So this is something very touching, that you can become friends in the society, not just colleagues, not just discussing science, but also sharing your journey in life, moments in life, things that you love. So I had the audacity of inviting somebody from England to an English tea service in Ann Arbor. So you can see Sylvia was approving of that English tea service. And Sylvia is always raising a glass for the success of others. And today, we raise a glass to the success of phenomenal, inspirational statistician, Professor Sylvia Richardson, and the person who brought us together, Professor Mike Boehnke. Please give them both a big round of applause. I know that Sylvia is anxious that she is losing minutes from her talk. She said that she needs 50 minutes, and we are definitely going to make sure that she starts on time. But Sylvia, I'm looking forward to your lecture. Every time I have heard you, you have inspired me, given new ideas, but also you're very comforting to the soul. Thank you very much for being a mentor.
Thank you so much. Yes, I've done it, I think. Thank you so much, Brahma, for this such wonderful introduction, and thank you to Michael. Is the sound okay? Yes, I think so. So as you could imagine, I'm extremely honored to have been the recipient of this prestigious award, and very pleased indeed that my name is now linked with that of Michael Boehnke, a giant in statistical genetics. So thank you so much for nominating, and the staff and everybody. And I've been here only a week. I came a week ago, but I feel like now I'm long-standing colleagues of all of you. So welcome I've been by staff and students alike, and I very much enjoyed all my conversation and all my meetings. And I hope I think we find every time something to exchange on different levels. So I want to particularly thank Brahma Mukherjee, my host, and because you can't imagine the careful planning that she has put. I mean, I think you can imagine because you know her. That she has put, her and Amanda, into making my visit both very pleasant but very fruitful and very diverse. And this is, I'm ever so grateful, Brahma. It's been, one of the best visits I've had in my life to a department, the best, I would say. And I'm also very happy to have had a chance now to meet Professor Boehnke, Michael Boehnke, personally, because of course I had heard about his work, but it is very different to actually meet the person. Statistical genetics, and human genetics, which is Michael's field, is actually at the root of the lecture that I will deliver today. And what I want to do is embrace and give, statistical approaches which can embrace both the statistical genetics and the genome data, but also a much more diverse set of data which looks downstream from the genome and which actually try to capture basically these downstream processes. And so that my talk is going to be about statistical approach to make the best use of this rich data world that we have now in biomedicine. So, right, everything works. So the health context, as we know, in which biomedicine is operating is really changing rapidly in sort of two dimensions. First, in the dimension that we can actually apprehend, collect data on very large number of individual or patients. Large cohorts have been set up, and for example, the UK Biobank has been really a worldwide example of forward planning, and now it's like a resources for the world to actually study the influence of genetics on sort of a range of phenotypes. Also, big international consortia have been focusing on specific disease, have been set up throughout the world. And Michael Boehnke is the founder on several of these important consortia. And that has again been a big effort from the community. And then we have also access to large database of electronic health record, and I know that Brahma and her team are really working on the analysis of that. So really, we have a large data collection, but also they are deep. Some of them, I mean, they're not necessarily both large and deep in the same time, but we also have now, because of biotechnology, which are advancing very fast, the ability to actually go in depth and probe into a mechanism. So it's called what we call multi-omics, going from the DNA to all the subsequent biological processes and capturing, gene expression, protein, and so on, and also large database of annotation. So this is a big challenge for any biostatistician, but for Bayesian biostatistician, because as Brahma was saying, I've worked not always, but most of my, more or less since the 90s, in Bayesian approaches, it challenges us to actually build kind of integrative models and hierarchical structure to actually jointly model some of those data sets with the aim to capture key features of underlying mechanism, but not in a clunky way. You also want to be able to, you can be ambitious in your style of modeling and try to do joint modeling, but you need to deliver scalable tools. Otherwise, the impact of your method is really lessened. So now it's really a challenge is you want to use the richness of the data, but you want to have scalability. And there's this kind of challenging task that I will try to show you some of the steps. So if we take a little step back now, we know that since the early 2000s, there's an ever-growing number of genetic variants which have been associated with a number of diseases. And every time I look at the GWAS catalog, I find, I have to update the slides, and in August it was over 500,000. I'm sure by now it's probably even more, and so on. And these are replicable results, and so they're key. They're indeed true results, but it's not necessarily enough to find, a genetic association, which could be very much in a non-coding part of the genome, to actually understand the functional effect. And this is where this additional data, this multi-omics data, can really help to actually follow the process through. And so it calls on us to develop methods to jointly model these data, the DNA and some of the downstream processes. So I would sort of categorize 3 types of common pervasive tasks in integrative genomics. One is, in the regression style, but it is to infer multivariate association, but between large set of predictors, the genome, and large set of responses, because we were not measuring one gene expression, we're measuring 20,000 gene expression. So we really want to do this multivariate association in both large predictor, large responses. We also try to get a first look at what is the structure, the multivariate structure of some of the data set, and particularly what are direct association between features. So we're interested in network inference, which is a first look at what goes with what, and what actually seems to be involved in the common mechanism or something. And then the third thing is basically to kind of to stratify populations, stratify groups of patients, and modeling the sources of heterogeneity that we see in the disease in a clinical, meaningful way. Now, since Friday, I gave a 2-hour workshop on clustering. I will not talk about clustering anymore today. You'll be glad to know, probably. But I will then focus on the first 2 topics. So the outline of my talk is there's basically 2 main areas. The first area, motivated basically by what's called expression QTL mapping, is a joint analysis of DNA, basically, but we could be large set of predictors, and large set of responses, and in particular to try to discover hotspots, i.e. region of the genome, which are associated with many responses. So not just within the gene, but very distal. And so there is an extension, so I will present that model, and it's an extension of this model which actually can incorporate additional covariates to try to understand more the functional presence of these hotspots. So these are the first two parts. And then I'll turn to the network and the task of actually trying to not only analyze a network, but a group of related data set, and try to compare and contrast their network. And that has led us to having this network, this joint network analysis has actually led us to use it to actually compare clinical groups, for example, responder and non-responder to a therapy, and try to, again, understand fundamental differences and possibly suggest new therapies. So the first pieces of work, I want to acknowledge two very close collaborators, Hélène Ruffieux and Leonardo Bottolo. Leonardo, I worked for many years on basically Bayesian variable selection and applied to eQTL mapping. And then Hélène, a very brilliant young woman from Switzerland, came fortunately to join our team, and she has been really instrumental in developing scalable, having an eye on the scalability, and also the actual technical knowledge to turn some of these complicated models into scalable versions and understand really their property. So the motivating application is, as I say, expression quantitative loci and the discovery of hotspots. I want to be able to jointly analyze many expression responses, in fact, all the expression responses, the 25,000 of them. So basically, and because you want to borrow information across these responses to get more power to discover these hotspots, which are variants, can be related and could be influencing, not in a proximal way, but in a distal way, mostly, the expression of many other genes. And so these hotspots are actually interesting but hard to discover, because if you just usually if the effects are not within, the variant is within the gene, the effects are smaller, are weaker, and then if you do many association and multiplicity correction, you more or less kill all these hotspots in one go. So it is really hard to find them, and this is why we wanted to develop this joint analysis. So that's why we call it hierarchical regression, because basically we will have some shared parameters which are specific to the SNP, which will be shared between all the responses to basically, which will be like a propensity for a particular variance to be a hotspot. And through this shared parameter, then you enhance the probability of actually detecting them. And as I was saying, we want scalable input. So we came up with a method which we call Atlas QTL, for detecting eQTL hotspot genome-wide, and here is like a cartoon of a cartoon. So the data set is basically on N patient, you've got a large genomic region, a few thousand SNPs in an LD block, for example, on a set of people. And then you've also got a very large set indexed by Q of responses, typically about 20,000 expression. And then we want to actually end up with a sort of joint map which tells us which SNPs are associated with which which expression, basically, which molecular traits. And this could be in cis, proximal, but most of them for the hotspot will be really distal. And then you can actually go along and collect these associations. And when you see these peaks, then you have hotspots. Now, we can't do, in terms of predictors, we cannot do the whole of the predictor and one, but we take large LD blocks and run basically in parallel. So a little bit of technicality in terms of, so there's a variable selection business going on, and there is some hierarchical sharing of information across the responses. So basically, in terms of notation, if YT is the response, so T index, the expressions, then we have a standard linear regression, but obviously we need to do some selection, and so we've got this coefficient. So S now is the SNP, the predictor, and we're supposing that only very few of those are actually significant. So we're putting a spike and slab model. That's pretty standard. And at the moment, this regression there is just indexed by the response, so there's nothing shared there. But the probability of having a non-zero association, so this probability gamma, is a Bernoulli, which is indexed by the actual SNP and the response. And it's in the modeling at the next level of this probability that we will actually share some information. So what model should we have? So instead of, you could run all of these in parallel, and this would not really work. But what you can do is, if you're looking for a hotspot, and there'll be few of them, you would want to have, it's like a propensity, an extra propensity parameter, which is shared, which says this variant has the propensity to actually control a number of expression or number of feature. And so we're modeling basically this omega, this probability of the non-zero regression coefficient, as with a column and a row effect, basically. So a SNP-specific parameter and a expression response-specific parameter. Now, let's look separately at these contributions. For the actual response, it's important because some genes have much more varied expression than others. So it's important to have a parameter which actually captures that and has a variance which would adapt to basically the chosen expectation for the prime number association. You need to control sparsity. So you need to basically more or less say that per response, you're not expecting too many SNPs being linked to them. You're expecting a few. So, and you could control that through these parameters, and you could adapt them to different response if necessary. Here we've chosen then to adapt them to basically the size of the LD block that you are considering. In terms of the interesting part is this propensity. So this kind of shared additional probability, well, not probability, but on the probability scale parameter, which sort of gives you a higher probability for a SNP to be a hotspot, so to control. So in a way, if you see it's lots of weak association piling up, this parameter will pick it up. The thing is, you are not expecting many hotspots. So you want, but so you want most of the time this parameter to be very small, to be nearly zero, really shrink that. But you nevertheless want sometimes it to be large. And to do that, you need a heavy tail distribution. And so we chose the famous horseshoe prior, which is such a flexible model because it's got both a global scale, which really shrinks most of these theta s, so the SNP-specific propensity will be shrunk to zero, but some of them will escape if there is enough information. So it doesn't really shrink everything, it just lets some. So it's called this multiplicative parameter, which will be a SNP-specific variant. I don't want to spend too long, basically, into explaining, the technicality of the model, because this is like now a horseshoe prior, but it's a horseshoe prior not directly on the beta coefficient, but on the probability. So at the next level, just want to say that it's important to have some kind of. You cannot, this global shrinkage parameter, you cannot fix it. It's very sensitive. If you take different value, you will get different results. And some value can induce large piling up of artifactual effect, basically. So you need some kind of consideration of how many responses you're actually analyzing. And in the paper, we do some detailed kind of discussion of why the Cauchy scale is now related, inversely related to the number of responses. So Q is the number of responses. So if you're analyzing 500 responses or 25,000, you're not expecting you will have a difference, theta zero, and this sigma zero, sorry, and this is important. So to demonstrate, so we've got an implementation. So this is, you can see it's a hierarchical model, 25,000 by 3,000 of variable selection linked. So this is quite fiendish, but you could do some variational inference. In fact, we've got annealed variational inference because, as you know, variable selection is a very multimodal. You could be stuck in modes, and it's good to be able to the algorithm to get out of some of the modes. So there's an annealed version of variational inference to actually implement this model, and this is an example which showing you that we're getting good recovery of the So there's 5 hotspots in regions where there's loads of LD, and we recover them well. But if we were not doing any annealed, because of the fact that there's quite a lot of LD and highly correlated predictors, you could get stuck in local mode. So this annealing version, or variational version, is important. Now, we've been working with some people in immunology who are interested basically in understanding monocytes, which are, white blood cells, very important for our fight against infection, bacteria, and so on. And so it's experimental data. They've got these monocyte expression, and then they act on these monocytes, and they stimulate them so that to see how the immune system would respond after an aggression. And so we analyzed basically 3 known quite large genomic regions, one on chromosome 12, one on chromosome 9, one on chromosome, sorry, I've put chromosome 12 twice. The first one is on chromosome 3, I think, 9 and 12. They had about 1,500 SNP each, and we did. So there's basically also unstimulated and then 4 stimulated condition. 3 stimulated condition, and then also some B cells. So that's why there's 15. And basically, we find that mostly that basically this on chromosome 12 in the Liz region, there is a persistent hotspot, and the colors depend whether it's for unstimulated, stimulated with IF and gamma, or different liposaccharide at different times. But basically, there is quite a high hotspot. In fact, they're not exactly at the same place. It's like a column of a hotspot there. So this is quite interesting, and it does rejoin some of the information that the previous analysis of this data had done. And with that, we worked with Ben Fairfax and his Oxford colleague. And so afterwards, we tried to see, so you find these hotspots, they make sense, but you want to carry on and interpret them more biologically. And you want to see whether there's some covariate, and in particular some annotation, epigenetic annotation, which actually could make you believe this hotspot is really doing something. And so we expanded the Atlas QTL to a sort of more confirmatory analysis of some regions. So whether Atlas QTL is really, you could basically just use the 100 LD block and run in parallel and then basically map across the genome. Here I'm talking about where we have found hotspot in a few regions. When you have found some hotspot, then you want to try to see whether some epigenetic process can be influencing these hotspots. So I'm talking about a sort of subsequent analysis, but on pre-selected regions. This is not for discovery, basically. So now you've got data as well on epigenetic marks. So the same data as before, and you've got on top of it epigenetic marks, and you want to see which, you don't want to, you want to in a way be agnostic which marks. You want to put a whole bag of possible marks and be agnostic and see whether some annotation actually are linked with the propensity of hotspot. And you could do that by expanding this Atlas QTL model. So at the first level, it's exactly the same model, but at the next level, then in this modeling, we had the propensity of being a hotspot, we had the response-specific parameter, and now we've got a regression on a bag of annotation, hundreds annotations. So here we actually try to be agnostic and take all the marks that you could get from ensembles, from EBI database, and so on, and add an extra layer of variable selection to be able to see which of the marks are influencing this particular hotspot. So I'm not going to detail the model. It becomes that we've got now a variable selection at another level again, but it actually does work if you have enough information. So you do need basically that the preselected region need to have enough information in there and need to have hotspots in there. If you just go genome-wide, you get nowhere. This model is too. But so this is basically the cartoon. So for this hotspot, we're now trying to find which of the epigenetic marks are actually increasing its propensity of hotspots. And when we did that on a hotspot in the Lys region on chromosome 12, we actually we put in variable selection of 168 annotation, and actually one came up, and that one was for monocyte specific. So it was really nice. Even though we put annotation related to other cell types, the sort of wide bag of annotation, the one which came up was monocyte specific. So basically, it comforts the idea that this hotspot is something is going on there. Okay. So now I'm going to go to the second part and talk about networks. And this work has been stimulated by the PhD thesis of Camilla Lingard, who is a very brilliant PhD student from Norway who just submitted her thesis in the summer. And she worked with me and Hélène to actually develop scalable methods for analyzing jointly multiple networks. So, we know that genes work together in complex systems, and we could capture how they work together by different types of molecular data. To get a first, if you wanted to model in detail, you would need the dynamics, and you could do some kind of mechanistic modeling of how genes work together. This is a much more superficial look, but a sort of first pass at the structure is to search for direct association between genes, i.e. just to find whether two genes actually have a direct association. I have a non-zero partial correlation, adjusting for the effect of all the other molecular entities in your network. So here in this plot, the genes will be, no, I'm going wrong. No, should be. The genes are the nodes, and the links represent a direct association, say, between these two nodes, conditional on all the other observed ones. That's what we're after. So, which are these, in contrast with the correlation networks, which just looks at the covariance matrix and actually just looks at the significance on the non-zero element of the covariance matrix, which are usually very full because there's both direct links and the indirect one created by their sharing a common child. So this, looking at direct association, gives sparser networks which are easier to interpret. But as you could see, it's an extremely high dimensional problem. Is that if you've got 1,000 nodes, which is not a very large network, you already got 500,000 edges, potential edges. So you need a lot of regularization to make this possible. You cannot, regress one thing. You have samples which are a few hundreds, where you want to regress one thing on more than 1,000 other things, so other nodes. So clearly you need regularization and shrinkage. So there's been the first tool that it's got some assumptions because you're basically looking at linear relationship. It's the Gaussian graphical models. And the Gaussian graphical models have been used extensively in lots of different biological literature. And there's a recent very nice review by me and Vera and other colleagues about the use of Bayesian graphical models for biological and molecular biology. Now, the work, which is the current work developed on network, people are trying to synthesize graphical information from related data sets. It's such a high dimensional problem, and you have small samples. Typically, you want to try to borrow information as much as you can. So if you've got related data set, you might want to see have they got common structure or not. There's also quite a lot of work going on trying to understand whether some of the links depend or are modulated by covariates. And then everybody is keen to have scalable tools because because of the high dimension of the problem. If you just have a pedestrian algorithm, it will take forever. And I'm going to talk mostly about the first point, which is synthesizing the information from related networks. Some previous work that I had done with a postdoc called Gwen Ledet we did something really quite simple. We adopted a conjugate formulation to the problem and choose an inverse Wishart prior for the covariance matrix. And because it's conjugate, you get all sorts of tractable posteriors. It's a lot of messy linear algebra, but you actually get nice explicit expression. And you end up having a kind of linear shrinkage estimator of the covariance, which is also invertible. So basically, you could also get an estimate of the precision matrix, the inverse. And we developed a simple way of testing, using the base factor for testing the presence or not of each edge. So it's not really a joint modeling afterwards. You can just see, okay, I have the base factor. I could test whether this edge exists versus it doesn't exist. So you have explicit expression and the null distribution. So it's extremely fast, and it gives you, so this is a tool where you've got very large network, you get something. But of course, it is quite constraining because it's linear shrinkage, and it's also it's not easy to consider multiple networks. So it's not so extendable to a richer framework. But if you need a first quick pass at if we have a very large network, it gives you an explicit answers and completely well justified, mathematically. So this immune hotspots that I was talking about across the different conditions. This is a typical example where you would like to try to do joint network inference, basically. You would like to have, non-stimulated, stimulated in 2 hours, stimulated and looked after 24 hours. You would like to see whether these networks are similar and pull information across and find the edges which are in common and the edges which are different. But most of the tools which were available when basically Camilla started her PhD, they were not feasible for the problem and the type of size problem we had, which was only 380 genes and a sample size of 300 or so. It just was not feasible. These methods could not really run on that type of data. So this is why we sort of looked to develop something more scalable. And because we liked horseshoe, and I think horseshoe priors is such a nice, it's got good statistical property. It's also amenable to quite nice computation and so on. We basically developed something which we've called joint graphical horseshoe, where we're going to model these related networks, but use a horseshoe prior to shrink the edges, to be pretty severe on the number of edges. So I will go and explain that now. I'll first talk about how we do it for one network, and then the extension to several networks will be quite straightforward. So basically, you've got just a network, so you've got a collection of data. You've got P nodes, P and then on N individuals, and you're modeling that as multi, is that my first slide? Yeah. You're modeling that as a multivariate normal with a covariance matrix. And in fact, what we are after is the inverse of this covariant matrix. But this inverse of this covariance, the inverse of the empirical covariance matrix is typically because you've got many, your n is small with respect to basically what you're trying, the number of elements. So you need some kind of shrinkage. And so modeling. So the hierarchical model that we define is to say that these elements, so theta ij is the element of the precision matrix between two nodes, I and j. And we are going to give it a prior, a horseshoe prior, basically. So you're expecting a few of these nodes, a few of these links to actually survive the shrinkage being there, and most of them to be zero. So there is, like usual, this global scale, which actually will make a lot of sparsity in your network, but you're having an ij element, a specific local shrinkage parameter, and this is the one where you're giving a heavy tail distribution. So some of these will be non-zero, basically. And it prevents over-shrinkage by having this Cauchy distribution with heavy tail. And this, so basically this Gaussian graphical model with horseshoe prior was proposed by Lee et al. In 2019, and they had a full, basically, Gibbs sampling implementation, which works well on and shows really good property, but on kind of modest size network. But as soon as you try with 200 known, it really grinds basically to a halt. So Camilla developed a variant of an EM algorithm to get the posterior mode, basically, of your precision matrix, which is, in fact, does an expectation step and some conditional maximization. So it isn't quite an EM, it's called an ECM, expectation conditional maximization. I don't think I want to go too much in the detail, but what happened is you introduce you reparameterize basically these lambdas which have Cauchy by introducing a set of, this is a classical way of dealing with Cauchy, another set of parameter, latent variables, the news, such that conditionally on the news, the lambda squares are inverse gamma, and these news are also inverse gamma. And because you do that, it's easy to take expectation. You get basically full conditional posteriors and of the news and of the lambda square. So you can actually do when you're doing your algorithm, you could do your E step, you integrate over these new and in an explicit way. So it allows you to do basically to compute what you need for your objective function in the E step, this reparameterization, and then you do alternate between maximizing the thetas and maximizing the lambdas. What is nice is that the convergence towards the posterior mode is fast, and so you've got good separation. Usually when you're having a horseshoe, you need some kind of threshold afterwards to distinguish. It shrinks, but it doesn't bring things to zero. It's not like a lasso thing. So you need then a threshold to say what is small and what is not small. But here, in fact, because we're having an optimization process, things really go very small and things just don't drop out, and there's clear separation. So there's no need to actually choose a threshold. What I haven't discussed yet is how we choose, because in the formulation that we have here, the global scale, I am not giving it a prior. And in fact, it is it's not possible to give it if you try to give it a prior and somehow things collapses, you get into some behavior which not you need to choose it following another criteria. You cannot treat it like the other parameter. And it's something which has been discussed quite extensively in the horseshoe, how to fix that scale. Sometimes people fix it thinking of some sparsity consideration. We decided to fix it in a more statistical way by using an archaic criterion for Gaussian graphical models. So we actually try to maximize the archaic criteria. And what's happening is that you take a grid of value from your global scale, and as the global scale, this tau square, it goes away from 0, we find that the archaic score basically stabilizes, and that's what we choose, how we choose to fix the value of this local scale. So this is an example of what happened when you choose different values of tau, and the archaic key actually stabilizes, and then we choose that. And it doesn't really matter after in the whole area afterwards. Basically, the network hardly changes, so you don't have to be too finicky. So this is what we've had called fast graphical horseshoe. So basically it's the same model as Lie, but implemented through this ECM algorithm rather than through fully Bayesian. And we did compare the output to the output of the Bayesian model for small networks, and we find very good correspondence. So it was nice to have actually a full Bayesian model we could compare to, and find that it was really more or less identical results. We are focusing on zero or non-zero edges. We're not focusing on the exact value of the theta. This is really an edge discovery and structural type of approach that you do. You're not really focusing there on the actual quantitative value of the theta. You want to see most edges disappear and what's left as association, and you're going to interpret this association as 0, 1, not as the value itself of the theta. So if you want to extend that to joint graphical horseshoe, then you have several networks. You have exactly the same setup, and the nice thing is that what you can do is you can, so here I'm actually just adding a k-index, meaning now it's the link between node I and j in the k network, which I'm giving a horseshoe, and the sparsity, the global scale is network specific, so this will stay indexed by k because some data sets are different. But for the lambdas, so the link specific parameters, if you look at their representation using this latent nu, then we can actually share. We are assuming that these lambdas, so these local scales, they come all from a common distribution which is not indexed by k. So this is how we pull information from the network. The edge-specific parameters all come from the same same prior, the same distribution, not indexed by k. That's at that level. So each network has its own kind of overall sparsity, but the edge-specific parameter are kind of parameterized with a global distribution, which is here. And so this parameter nu ij is interpretable in the way that if it is 1 over nu j is away from 0, it will say there is evidence that an edge is shared. And this is like an indicator that an edge is shared. And otherwise, if it is very much 0, meaning there are very specific edges. So to show you what happened here, for example, we have like two networks with 40% shared edges. there's 100, the number of nodes is 100, and the number of samples is, for one sample is 100, and the other is 150. And then if you see here, we have got basically, so the truth is, if it's like a circle, this would be an edge which is shared in both networks, and if they are either squares or triangle, they're specific to network 1 or network 2. And you can see there's a good discrimination of the truth versus the others. The theta is actually 0. And you could also see that the new inverse, this parameter nu ij, is, here it's 0 for all these squares or triangles which are specific to each networks, and these are the ones which are, which are shared, the edges which are shared. So basically, there's a good discrimination between the common edges and the specific edges that you could see with this new IJ, and a good recovery. Lou, how long have I got now? Another 20 minutes? Okay, that's okay. So I could show you now. Basically, we try to see what, is it interesting to use this joint versus analyzing each network separately, which obviously you can do. And so we did a cell simulation study where you had from 2 to 10 networks, and with different proportion of similar edges going from all their edges where. So this is the disagreement scale. So there, all the edges, there's no disagreement. They all have similar edges. There are no common edges. And this is when we had 2 networks, 4 networks, or 10 networks. And we run either a joint network analysis, or you could run the network separate and then look at basically the recovery in terms of precision. So the positive predictive value in terms of basically among those who if you've declared an edge existing, it is the probability that is actually a true edge. And that's what's the precision of positive predictive value. And you could see that, of course, the precision is higher when all the edges are in common when you're using. So this is in this green solid line is the joint, and in the dotted line is the separate analysis. So you do, when there's a lot of edges in common, you do better by using the joint network. As the disagreement grows, you're doing less and less well, and you more or less do the same. Sometimes, surprisingly here, you seem that even when they disagree totally, you seem to be doing better. It's not you seem, you are doing better by using the joint network analysis, even though there's no edges in common. But what there is in common is when a lot of non-edges. And in fact, that by using this joint and saying you're sharing the nu I, even if the nu I is going to be 0, basically, it is propagating some information about the non-edges. So somehow you don't lose anything by using the joint graphic. It will not tell you you've got common edges, but it will stabilize the non-edges. The noise. So that is quite nice. We've compared with several lasso type techniques for doing these Gaussian graphical model, in particularly a technique which is called SPICE and SLAB joint graphical lasso. Because the joint graphical lasso, as it was originally defined, really over-select the edges because their criteria for selecting the edge is not well calibrated, and I think that has now been recognized. And this spike and slab geographical lasso tried to have a better criteria for selecting the edge, and you could see the results. The results are similar. What we find is, so here again you've got in the circle are the edges which are present in both, and we see we detect them well. Geographical slack and slab geographical lasso also detect them well. So some confusion, but it's less of a clearer picture because somehow it's kind of really trying to push the networks together in a strong way. It selects more edges, and you could see those which should be just for one of the networks, they're getting also pulled towards the other network. So there is more confusion. There's more really forcing the network together if you do this, John. So it's fairly comparable, but we find that we estimate better, separate better the edges which are specific to each network from the common one, because the horseshoe is really, letting that freedom happen. We also compare in terms of precision and so on. The simple geographical lasso has very poor precision because over-selects completely the edges. It's got very high sensitivity, so it finds everything, but it finds also lots of false edges. Whether basically the spike and slab geographical lasso and us, we are on par in terms of the result. We have better, when, as I was saying, when the network disagree with each other, we have better precision than this geographical lasso, which really shrinks towards a common and kind of overshrink towards a common. So it emphasizes too much what's common, not. So there's many in the paper, which is an archive. There's extensive simulation. Now we come back to what we wanted to do: look at the immune system. So if you remember our first eQTL, we got hotspots and in this sort of unstimulated and these stimulated conditions. And now we want to see, are these networks, we could take this set of genes and we could make a meta set of genes and then look at whether these networks are similar or not, across these conditions. So first we basically we look at like 381 genes associated with the hotspot in Liz in this chromosome 12 SNP in at least one condition, but we could also focus, so this is like the union of all the genes which were associated with the hotspot in unstimulated or stimulated at 2 hours, 24 hours, and so on. So you could also look specifically at the networks, the subgraphs of this smaller set of genes, and see how they are distributed in the large graph. And we look specifically also around neighbor of Liz. So this is a big. It's not so fast to run. This is a big. It's a big analysis. 4 conditions, 2 samples of size 200 and 300. These are large networks. So this plot there, you could look at it for a long time, but we've done some kind of histogram and tables. There's 4 conditions, so there's 4 symbols, but basically. About a third of the edges are in common, which is quite something. If you took randomly, random, you would not get that. It's very sparse. It's about 2% of edges which survive. But what was interesting was that the gene controls by the hotspots in each condition, you could test by permutation that they were denser. And also that in this genes controlled by the hotspot, if you look at Liz, because Liz, the question is this gene Liz, how is it controlling this whole network, or is it helping to mediate something? You just try to understand what is the role of Liz in this process. And what was interesting, and possibly pushed towards Liz having a mediating effect, is that the neighbors of the Liz in the network were enriching links to the hotspot. So somehow there's a hotspot, there's Liz, and they have many links. But we also discovered new hub genes. So in these networks, there are some genes which have many links, and our collaborators are looking at those which were not particularly known. So these other features actually come in. So there could be other drivers for immune response. So the last thing I want to talk about is an application to some clinical trial data where we have used this comparison of network to basically compare groups of responders and non-responders to therapy. And somehow developed a strategy of first doing the network analysis and then looking what for in the networks that you get, whether they enrich for some gene sets or pathways. I mean, the thing is that because you can analyze jointly these networks, you can compare them. You can actually, because you've put them on the same footing, basically. You could, if you analyze separately the networks, the sparsity will be so. There won't be, they haven't been in the same framework. They will be less comparable because some could be very sparse and some could be less sparse and so on. So I think even if you want to see differences, it's important to start by putting them in the joint framework. So what you could do is you got basically, you jointly estimate the networks, then you could rank the genes or the proteins. I mean, it's not necessarily expression, it's anything. You could rank the protein using their node degree. So you have in each of the network, you could have the hubs and the subhubs and etc. And then you could look at different pathways and see whether the hubs, you see whether the important genes are belonging to a pathway or another pathway and so on. So this is what we've called basically enrichment strategy for network comparison, which is which I've tried to illustrate here. So first you've got your data and you actually estimate these networks, and then you rank the genes in each of the set following how many links there are, and then you could use basically the gene enrichment strategy on that. And so we've been working with colleagues in Oslo, Mats Haugen and Olaf Engebrat, who are PI on an important clinical trial on breast cancer. It's a phase 2 clinical trial with patients with large HER2 negative, so difficult patients, not the standard breast cancer patient which have some poorer prognostic. And they were randomly assigned to treatment with either the usual neoadjuvant chemotherapy or in combination with bevacizumab, and I will say Bev afterwards because it's quite a mouthful, but an immune, basically, an immune therapy, which was you try to see whether it could enhance the effect of the chemotherapy. And because it obstructs the formation of blood vessel, and so, they thought this drug was tried for this particular type of difficult breast cancer patient. And this is a very, very rich trial because they measured all sorts of additional data besides the imaging and the response and monitoring. They also measured proteins before the treatment started, during the treatment, and at the end. And in the end, they also, of course, assessed whether there was response or not, pathological complete response or not. So in the treatment arm, there was, in fact, only 14 responders and 14 non-responders. So these, if we wanted to compare, to try to see whether the biological structures of the network of these proteins is different between the responders and non-responders, is really small. 14 versus 40 is too small. But fortunately, that team had also identified a sort of predictive signature based on 9 proteins, which predicted treatment response. So, so we could use basically this predictive signature to sort of predict the response in the control arm, which was not treated. So it would be what, we were predicting what these patients non-treated, which would be the responders and the non-responders. And so that gave us more. That meant we could use the whole cohort, and that gave us numbers of which we could actually do network analysis. So basically, we did now a comparison of the network between these 40 responders, including the predicted responders, and the 76 predicted non-responders. And we looked at differences between the protein networks and different pathway. We found that basically the networks were really quite different. There was only 120 edge in common, and in fact, it's significantly different with respect to a permutation test. We find that if we did a GSEA on the responder network, we found 3 pathways which were of sizable gene sets which were enriched, and we could reproduce using TGCA data. So we actually reproduce this enrichment in an independent data set. In the non-responders, there was nothing, no significant gene set emerged, even though it was a bigger group. And we hypothesized it's because de facto, it's a much more heterogeneous group because they haven't responded. So they don't share anything in common. And so we stratified again by an inflammation score, a tumor inflammation score, which has been proposed as something important in cancer. And I'm sure Jeremy and other people know more. But they were suggested by a collaborator. And then we had 19. So among the non-responders, we now had 2 subgroups, and we compared these networks. And currently, then some pathways have come up, and some interesting things have come up. And currently, our collaborators are deciding whether to actually test some of these target pathways which have come up in mouse model or something, just to try to validate. So they're quite excited by the results, and we're very pleased that this tool, which is quite sophisticated and so on, seems quite far-fetched from reality, seems to be actually giving really potential targets. So I'm arriving to my concluding remarks, which I think I've explained that I think this type of Bayesian multivariate modeling and scalable, accompanied by scalable computation, just have a lot to offer to develop, to increase biological understanding. My take has been to try to get away from one-at-a-time analysis and really tackle the multivariate aspect and try to learn from that. Of course, it comes at computational cost, and it's not always possible. And one way to really use the data fully is to try to borrow information. So in this eQTL model, we borrowed information across, weak evidence across many responses was pulled up to find this hotspot. In the network, again, you try because you try to borrow information across several networks or compare several networks to get interesting information to come up. So that can give a strategy like hotspot discovery and a gene set associated to the hotspot, then you do a network analysis on there, and you might find, some new immune driver. Or you contrasting clinical group, you do joint networks, and you try to see if they're by ranking, the nodes by how active they are in the networks, if you could find differential pathway highlighted in the groups to try to understand how they're different and things like that. All that is complementary to the traditional analysis. So I just want to thank again my close collaborators, who are Elaine, Leonardo, and Camilla, and it's been such a joy working with them, and also Brahma, who unfortunately is with the provost at the moment, and so she can't hear my thanks, but Michael can hear my thanks, and the whole of the department, and I see many of you here for your invitation, splendid welcome, and it's made my stay very memorable. Thank you. Yes, please. And here I've left some references, but you could always also email me if you want to discuss further.
Yeah, hi. So your first presentation ran into like an item response theory model. You could sort of break apart this subject love or the sort of
Yes.
molecular component versus the SNP component, right? And you might think of the molecular components.
Yes, I mean, so this probability was, yes.
So I guess that assumes there's no interaction. But also, I mean, if you sort of go down a response theory model, there's some sort of variability in individual performance, right? Is there a reason to think that would be the case with the SNPs, that some kinds of SNPs are more likely to be going across multiple?
Yes, yeah, I think, that.
I'm not a expert.
Yes, I'm not a virus, but I mean, I think people do believe that there are some region or so.
The model allows you to go to zero if you believe it there, right?
Yeah, to actually, because basically inside the gene, you've got variation inside the gene you are, then it's expected it would have an impact. You could understand that mechanically, but it's the way that it could influence much further. It's more complicated, but people believe there are some true, there's all the spatial aspects of how it communicates in the genome. There's a lot of debate about how these things can happen, but there are definitely biological mechanisms where some loci can be key drivers of process. But they're hard to find, and they're hard to find, and most of the eQTL have been done, one at a time, and only looked at within gene, the variants within the gene which really influence the protein expression. So because these are stronger effects, and in a way, they're easier to understand. So you could think the others they are weaker. So you tend to, since you're testing, if you do that one at a time, you're testing so many things that you have to also really threshold that and a lot that disappear. So I'm really hoping that we will have the energy now to really pursue. I think these are tools. They're good tools, and they need to be very good to use them in very meaningful data set to actually really map and across the genome for specific types of cell and try to interpret those, further experiment, and so on. It would be great to have collaboration with, the immunologists in some areas of biology where people are really interested to map. And I've talked about expression of genes, but it could be used for proteins just the same. You don't have, I think, people moving in a way from expression towards protein, direct measure, and this would be ideal for protein as well. So you could do pQTL, and it doesn't have to be eQTL, or it could be for metabolites or whatever. Vera, Jeremy. Sorry, Lou, I'm Manchester.
No, real quick, more of a clarification. Like, how do you do enrichment analysis with networks? Like, how does...
Sorry?
How do you do enrichment analysis with networks? Usually, it's some level of group testing, right? That
yes, I mean, we did rank we ranked the feature by, you need to get a set of feature and rank them, and then you need to see whether the enrichment. I mean, that's what GSEA. So we rank them by their degree. Yeah, so in a way, meaning how active they are, or how, how influential, not necessarily active, but how influential they seem to be. So we rank them by the degree, but you could have other ranking, I presume. And then we just did the standard GSEA, but there could be some other tools. But I think you've also done. I mean, somehow the degree of is quite a good way to think of that these nodes are more relevant somehow, not less isolated. So in a way, but I think there could be different ways of different scores that you could use. But there we did the simplest, which was rank them by their node degree and then do enrichment analysis on that. For the classical set of pathways,
I don't have a lot of experience with variational inference, but we had a student who graduated earlier this year who was looking at longitudinal data and using horseshoe priors. And it was a situation where you could do a full MCMC and you could do a variational thing. And the variational inference was like orders of magnitude faster, I mean, hundreds of times faster. But for posterior distributions that estimated weren't always that lined up with the full ones. It seemed to find the right places when these horseshoe parameters should be like really non-zero. It did a good job at that, but sometimes the posterior distributions didn't line up that well, and maybe that doesn't matter. If you don't want to pretend you're being fully Bayesian, you've just got an algorithm for fitting the data, and it fit the data quite well. But I don't know if you have a similar experience with that.
Well, I think in this particular context, in the context of the in a way, in this variable selection context, we're more interested in the, the detection than the actual quantification in a way. So I think this is, I agree that, posteriors, it's an approximate thing, and the posteriors will not necessarily line up well. But there, in terms of getting, the key variables or not, it was doing a very good job. But if you are using variational Bayes to actually really model, you need the full posterior and quantitative. I think indeed you'll find probably, it really depends what's your aim. And for our situation, it was well suited, and to give, to be able to select the feature, select the hotspot or select the edges. well, we didn't use variational base, we used another approximation, but it would be the same. You're getting just the posterior mode and some measure around it, but it's not the full posterior for sure. Yeah. And I think you don't necessarily need to do mean field. I mean, there's different ways you could choose following the problem at hand. So if you've got correlated data, you might, I don't know how you defined your variational approximation, but you could try to not choose, you wouldn't just make them independent. You try maybe blockade or some kind of closer approximation if you understand the structure of the data, which is a bit less rough than just the basic mean field approximation.
Some of those things, but I guess if it doesn't give you the right inference from a Bayesian perspective, that you could still trust the variable selection, you think? Okay.
But you can get stuck. I mean, it's more that you could trust the variable selection, but you have to be careful not to be stuck in like all these optimization. And so that's more the concern, and that's why we were doing this, and we have to do this anneal version for variable selection. It really helps. It's very much like people who do EM for variable selection. They really also have to be careful about not being stuck and restart. So yeah. So in terms of comparing performance, it was doing really well if you use the anneal version. Otherwise, you weren't getting the right answer.
All right, so the reception actually officially already started. I know you may have other questions, but the good thing is Sylvia will be around. So feel free to ask her questions during the reception. So with that, let's thank our speaker again. Thank you so much.
Thank you very much. You're welcome. And thank you for coming here. And look forward to the reception.